Hi, I'm Yasunori Toshimitsu from the University of Tokyo, and I'm here to present our paper for IROS 2020, Biomimetic Control Scheme for Musculoskeletal Humanoids based on the motor directional tuning in the brain. We have used musculoskeletal humanoids in this research. These robots have a passive skeletal structure with actuators that emulate muscles. We consider that such a biomimetic design entails two potential benefits. First, it could achieve more human and animal-like movement, so it could lead to breakthroughs that will lead to more capable robots. Also, these can be used as a human body simulator platform, which can be used to test out hypotheses regarding human motor control. Thus, through these robots, we could understand biological motor control. In this research, we have attempted to control a musculoskeletal humanoid based on algorithms underlying human motor command generation. Next, let me give a brief overview of the motor command pathway. High-level commands are issued from the cerebellum and the basal ganglia, which are then processed through the motor cortex and the brainstem centers. They are finally converted to commands that lead to individual muscles in the lower-level circuits in the spinal cord and brainstem. The motor directional tuning phenomenon, which we have attempted to emulate in this research, occurs here in the motor cortex. Here it is important to note that the representation at the motor cortex is more abstract than the individual muscle level. Here's an overview of the motor directional tuning phenomenon in the human motor cortex. Neurons that exhibit the motor directional tuning are activated in an orderly fashion based on the direction of our movement. For each of these cells, we can measure the directional tuning profile, which is the activity of the cell plotted against the movement direction. In the left graphic, as an example, 3 directional tuning profiles are shown for 3 cells. The preferred direction of each cell is another important element in motor directional tuning. It is the movement direction in which the cell is most active. For example, for this cell, the preferred direction is an angle, the movement direction of angle 0. And for this cell, the preferred direction is the movement direction at an angle of pi. So for example, if the hand moves in this direction, each cell is activated according to its DTP, and thus for the activation is the highest for this cell. Here's the proposed controller based on the motor directional tuning phenomenon. From the eye camera image, using the AR markers, the object poses are detected. Then the movement direction is calculated. This is calculated based on the task that is at hand. For reaching task, it'll be the vector from the hand to the target. Then the movement direction vector is given to the controller, and for each muscle, the attention command is calculated through this process. First, the angle between the movement direction and the muscle's preferred direction is calculated. Then the activation of that muscle is calculated through the directional tuning profile, and then finally the tension command is given through the low-pass filter. The tension commands are given to the robot, and then the robot is actuated. This process continually runs while the robot is moving, and thus the robot can adapt to changes in its body pose and the environment. Here, let me clarify the differences between the original motor direction tuning phenomenon, which is the neuroscientific phenomenon, and our controller. Although the basic principles are the same, the implementation has been simplified for our purposes. First, while there are hundreds of cells in the motor cortex that exhibit motor directional tuning, here in our implementation, there is the same number of cells as the same number of muscles. The relationship between each cell and muscle is different as well. Whereas in the original phenomenon, each cell activates multiple muscles. And the motor cortex signals are converted into muscle commands as it travels through the spinal cord. Therefore, it is not a straightforward mapping between the cell and the muscle. On the other hand, on our controller, we have a simple one-on-one relationship where each cell corresponds to a single muscle. Also, a gradual change in preferred direction has been recorded as arm poses changed, but in our implementation, the muscle preferred direction does not change across all arm poses. Next, let me explain how we have obtained the muscle preferred direction. We have obtained the preferred direction for each muscle after holding the arm still in a neutral pose like this, and then increasing the tension for each muscle, and then recording the resulting arm movement. The muscle's preferred direction is set as the normalized movement direction. By characterizing on the real robot rather than a simulated model, we do not have to worry about modeling errors that result from the complex musculoskeletal structure. This is the resulting muscle preferred direction. Next, we have evaluated the proposed controller in a reaching task. Here, the robot tries to reach its hand to a target in front of its body. For comparison, we also did the same experiment using a controller based on the geometric musculoskeletal model. Here, the muscle length is calculated from the musculoskeletal robot and is sent to the motors. The model pose is updated continuously to minimize the error between the hand and target. Here's the result. The top graph is the distance between the hand and target, and the lower graph is the muscle tension. Both controllers were able to gradually adjust its arm pose to reach the target. However, note that due to the model error, the differences between the actual robot and the model pose increases as the geometric model-based controller reaches closer to the target. Next, let's take a closer look at the tension history of muscles involved in elbow movement. Here, the agonist muscles which generate motion are the muscles used in elbow flexion, muscle ID 18 and 19. We can see that in the proposed method, there was coordinated muscle activation across the agonist muscles, and these muscles were able to work together to generate the elbow flexion movement. The antagonist muscle, which is the muscle involved in elbow extension, has been kept low. However, for the comparison method, the model error results in large internal tension, and the muscle tension for the antagonist muscle increases as well. Furthermore, the coordination of the agonist muscles is lost. As you can see, for muscle ID 18, which should be being used to generate motion, the muscle tension is decreased as time goes by. As a result, as for the reaching accuracy, it was better on the comparison method since it was able to reach closer to the target. On the other hand, for muscle tension, the overall muscle tension was kept lower for the proposed method, which was able to use the agonist muscles effectively. Next, we have applied the proposed controller to neck muscle control. The eyeball controller follows the visual target and also keeps the eye orientation still during head movements. The head controller uses the motor directional tuning-based control, and it controls the neck muscles to align the head with the eye orientation. The two controllers are coupled only by the eyeball joint angle pose, but are able to work together to create coordinated target-following movement. Here it is applied to a visual following task. You can see that it first tracks the target with its eyes, and then the head catches up to align the head with the eyes. Finally, the three controllers for the eye, neck, and arm were run simultaneously to generate a coordinated upper body motion that tries to track a visual target with the arm and head. In conclusion, we have created a biomimetic controller based on the motor directional tuning phenomenon that was applied to the control of the arm and neck muscles of a musculoskeletal humanoid. It uses a simple sensory motor mapping instead of an internal musculoskeletal model. This sensory motor mapping continuously monitors the relationship between the body and the environment and adapts its output accordingly. We believe that improving the fidelity of the sensory motor mapping, which is the mapping between the desired movement direction and the muscle activation, can improve accuracy and range, which were found to be limiting in the experiments using this controller. Thank you for listening.